Let us begin our journey into the world of bone anatomy, because before we can fully appreciate the intricacies of the foot, we must first understand the fundamental building block of the skeleton itself. When you look at this page, you can see a long bone illustrated on the left side, with its major regions carefully labeled. Imagine holding a femur or a tibia in your hand. At the ends, you find the epiphyses, the rounded extremities that usually form joints with other bones. Just beneath that lies the growth plate, also known as the physis, which is a crucial zone during development because it allows the bone to lengthen as a child grows. Between the physis and the shaft sits the metaphysis, a region rich in trabecular bone, where much of the dynamic activity of bone remodeling takes place. The shaft, or diaphysis, is the long central part, primarily made of dense cortical bone that gives the bone its incredible strength. And finally, when the bone extends into the articular cartilage of a joint, we refer to that part as interarticular. Now, notice the small circular sections shown on the right. When we cut through the shaft, we see a thick wall of cortical bone, hard and compact, designed to resist bending and protect the inner marrow cavity. In contrast, when we cut through the metaphysis, the architecture looks entirely different. Here we find cancellous bone, also called spongy bone, which appears as a lattice or honeycomb. This arrangement may look delicate, but it is remarkably strong while also being light, and it provides space for bone marrow where blood cells are produced. Zooming in even further, the diagram of cancellous bone shows us a living tissue in constant activity. The trabeculae are thin struts of bone arranged according to the mechanical stresses placed upon them, almost like the beams of a bridge adapting to weight. Lining these trabeculae are osteoblasts, the bone-forming cells. Some are active, secreting osteoid, which is the unmineralized organic matrix that later becomes hard bone. Others are inactive, resting as lining cells, waiting to be called back into action. Embedded within the bone itself, in tiny lacunae, are osteocytes, the mature bone cells that maintain the tissue and sense mechanical strain, communicating with one another through delicate cannuliculi. And on the surface, we find osteoclasts, large multinucleated cells that resorb bone, creating little pits known as houseships lacunae. This balance between osteoblasts and osteoclasts is what allows bone to continuously remodel, adapt, and repair itself. If we shift to the table below, the essential functions of bone are summarized. Beyond simply forming the skeleton, bone is a dynamic organ. It serves as an attachment site for muscles, enabling movement. It shields vital organs, such as the cranium protecting the brain, or the ribs encasing the lungs and heart. It also functions as a reservoir for minerals. Remarkably, 99% of the body's calcium is stored as hydroxyapatite crystals within the bone matrix. And perhaps most crucial for life, bone houses the marrow, the site of hematopoiesis, where blood cells are born. Bones come in different forms. Long bones, like those in the limbs, develop mostly by endochondral ossification, meaning they first form from a cartilage model. The only exception is the clavicle. These long bones have primary growth centers in the shaft and secondary centers in the ends. They are divided into three parts: the diaphysis, with its thick cortical wall surrounding marrow, the metaphysis, with its cancellous architecture, and the epiphysis, which often contributes to joint surfaces. Flat bones, in contrast, such as those of the skull or pelvis, form by intramembranous ossification, directly from mesenchymal tissue without a cartilage stage. At the microscopic level, we can distinguish woven bone and lamellar bone. Woven bone is immature or pathologic. Its fibers are irregularly organized, and it is not well adapted to handle mechanical stress. You see it in infants, in fracture callus, or in certain tumors. Lamellar bone, on the other hand, is the mature, highly organized form, built with precise orientation to resist stress. Both cortical bone and cancellous bone, once matured, are made of lamellar bone. So, as you reflect on this page, think of bone not as a dead scaffold, but as a living dynamic organ, strong yet adaptable, protective yet interactive, and essential for movement, mineral balance, and blood formation. This foundation will guide us as we move deeper into the study of the foot, where these principles are beautifully expressed in the architecture of its many bones.
Now let us take a closer look at cortical or compact bone, which is beautifully illustrated on this page. At first glance, it may seem like a solid, impenetrable structure, but when magnified, we see an intricate, highly organized system designed for strength, nourishment, and constant renewal. Starting from the outermost surface, the periosteum covers the bone. This thin but vital membrane is richly supplied with vessels and nerves, and it serves as the entry point for blood into the dense cortical wall. Just beneath the periosteum, we find the outer circumferential lamellae, layers of bone running parallel to the surface, giving stability to the entire shaft. From here, small periosteal vessels penetrate inward, linking up with deeper channels. Running through the cortical bone are haversian canals, each containing capillaries, nerves, and progenitor cells. These canals are the lifelines of the bone, ensuring that even the innermost osteocytes receive nutrition. Perpendicular to them are the Volkmann's canals, which act like cross streets connecting one haversian canal to another, and also linking periosteal vessels on the outside to the deeper network within. This three-dimensional vascular system is what keeps bone alive and responsive. In the center of the shaft, the nutrient artery enters through a foramen in the diaphysis, travels inward, and then divides into branches that supply the marrow and the inner cortical layers. Notice how the diagram shows the nutrient artery giving rise to smaller arterioles that enter Volkmann's canals, ensuring even distribution throughout the compact bone. Eventually, these vessels anastomose with metaphyseal arteries near the ends of the bone, creating a rich collateral network. Within the compact bone itself, the structural unit is the osteon, also called the haversian system. In the enlarged diagram at the bottom, you can see 6 concentric lamellae arranged around a central canal. Think of this as a series of tree rings, each representing bone laid down at a different stage. The central canal houses a capillary, a nerve fiber, and supporting cells, while the lamellae are made of collagen fibers aligned in alternating orientations, which greatly increases strength. Between these lamellae sit osteocytes, the mature bone cells, each within its own lacuna. They extend tiny cytoplasmic processes through channels called canaliculi, connecting cell to cell in a living communication network. This allows nutrients, waste, and signaling molecules to pass between osteocytes, ensuring that the bone behaves as a coordinated organ. At the periphery of each osteon lies the cement line, a boundary that marks where bone resorption stopped and new deposition began. It is the scar of a remodeling event, a record of the bone's dynamic history. Over time, as osteons are replaced, fragments of older ones remain as interstitial lamellae, filling the spaces between new systems. Deep inside, we find the inner circumferential lamellae lining the endosteal surface, facing the marrow cavity, where trabeculi of cancellous bone project inward. If we now compare compact and cancellous bone, as summarized in the table, cortical bone is the powerhouse of strength. It makes up about 80% of the skeleton, is thickest in the diaphysis of long bones, and is designed to resist bending and torsion. It grows slowly, remodels at a moderate rate, and forms the rigid shell of our skeleton. By contrast, cancellous bone is lighter,
arranged in a delicate lattice, and though it accounts for only about 20% of skeletal mass, it has a much higher turnover rate. This makes it more metabolically active, but also more vulnerable to conditions like osteoporosis, where trabeculi thin and fractures become more likely, especially in the vertebral bodies, femoral neck, and tibial plateau. So when you imagine compact bone, think not of a lifeless wall, but of a bustling city, arteries and canals supplying nourishment, osteons forming strong concentric neighborhoods, and osteocytes whispering signals through their canaliculi. This microscopic harmony is what gives the macroscopic skeleton its remarkable durability and adaptability.
Let us now move deeper into the very substance of bone itself, its composition. Up to this point, we have spoken of macroscopic features like the diaphysis, metaphysis, and osteons. But the true strength and resilience of bone comes from the microscopic balance between its organic and inorganic phases, a marriage of flexibility and rigidity that makes bone unique among tissues. If you look at the main illustration, the organic component, which makes up about 35 to 40% of bone by weight, is represented in yellow. This phase is primarily matrix, and within it, collagen reigns supreme, accounting for roughly 95% of the organic fraction. Collagen fibers are carefully aligned, and they give bone its tensile strength, the ability to resist stretching and pulling forces. Imagine a rope woven of countless fine threads. This is the role of collagen in bone. Within this organic phase, we also find proteoglycans and non-collagenous proteins. Proteoglycans are fascinating macromolecules built around a hyaluronic acid backbone with glycosaminoglycan chains branching out like bristles of a brush. They occupy space, retain water, and influence mineralization. Non-collagenous proteins such as osteocalcin, osteonectin, and osteopontin play important roles in bone turnover and in binding cells to the matrix. Osteocalcin, for example, is often used clinically as a marker of bone formation and turnover. And of course, embedded within this organic matrix are the bone cells themselves. Osteoblasts, which originate from mesenchyme and lay down new matrix. Osteocytes, which are mature osteoblasts trapped within lacunae and serve as mechanosensors. And osteoclasts, which come from the monocyte macrophage lineage and resorb bone. Together, these cells orchestrate the constant remodeling that keeps the skeleton healthy. Overlaying this framework is the inorganic component, shown here as the mineralized matrix. This makes up about 60% of bone weight. The dominant mineral is hydroxyapatite, a crystalline form of calcium phosphate with the formula Ca10PO46OH2. These crystals deposit between and at the ends of collagen fibers, stiffening the structure. While collagen provides tensile strength, hydroxyapatite provides compressive strength, the ability to resist weight and pressure. This duality, collagen plus mineral, explains why bone can be both strong and slightly flexible, rather than brittle like pure mineral or soft like pure collagen. A minor contributor to the inorganic phase is osteocalcium phosphate, sometimes called bruchite, which also plays a role in mineralization. If we zoom in to the molecular level, the diagram of collagen shows the alpha chains that make up type 1 collagen. Each chain is about 1,000 amino acids long, and every third amino acid is glycine, the smallest amino acid, which allows the chains to pack tightly into a triple helix. 2 alpha-1 chains and 1 alpha-2 chain twist together, forming fibrils, which then bundle into fibers. In bone, as in tendon and ligament, type 1 collagen is the dominant form.
The proteoglycan structure shown to the right illustrates how these molecules are built. At the core sits a protein, with branches of chondroitin sulfate and keratin sulfate extending outward. These branches are glycosaminoglycans, or GAGs. They link to a hyaluronic acid backbone via special link proteins, forming enormous aggregates. These aggregates contribute not just to compressive strength, but also to the regulation of mineral deposition within the collagen network. Finally, we must not forget water, which makes up about 5% of bone by weight. Though it seems small, water is critical to the diffusion of nutrients and the flexibility of the matrix. Its proportion varies depending on age and location. At the very periphery, the periosteum surrounds the bone. It is a vascular fibrous layer thicker in children, where it actively contributes to bone growth, particularly in the diameter or width of long bones. Clinically, this explains why children's bones heal more quickly and why periosteal reactions are often seen on radiographs after injury. So when we think of bone composition, picture it as a carefully engineered composite material. The organic collagen fibers provide the ropes, the proteoglycans and proteins form the scaffolding, the hydroxyapatite crystals stiffen the whole structure, and water ensures flexibility. This balance allows bone to withstand the stresses of life, remodel itself throughout decades, and serve as the central pillar of the musculoskeletal system.
Now that we understand the composition of bone, let us turn to how it is regulated, how bone mass is gained, lost, or maintained throughout life. This page illustrates beautifully the balance between deposition by osteoblasts and withdrawal by osteoclasts, and how various factors can tip the scale toward bone gain or bone loss. At the center of the image, you can see a pile of bone mass, like sand being poured into or taken away from a container. The level of this pile reflects the net state of bone, whether it is increasing, stable, or decreasing. On one side, the forces of deposition predominates. On the other, the forces of resorption take over. The balance depends on the activity of osteoblasts, the bone-building cells, and osteoclasts, the bone-resorbing cells. Let us first look at stimulation of deposition, the first mechanism shown on the left. Here, osteoblasts are numerous and active, laying down new matrix. What drives this? Weight-bearing activity, such as walking, running, or resistance training, is a powerful stimulus. Growth in childhood and adolescence also increases deposition, as does fluoride or even certain forms of electrical stimulation applied to bone. At the same time, osteoclasts are fewer or less active, meaning bone resorption is reduced. The result is a net increase in bone mass. This explains why physical activity is so important for building strong bones, especially during youth. Now, contrast this with inhibition of deposition, the second mechanism. In this situation, osteoblasts are fewer or sluggish, so they deposit less new bone. This occurs with lack of weight-bearing activity, as in immobilization or prolonged bed rest. Chronic malnutrition, alcoholism, and many chronic diseases have the same effect, as does normal aging and states of high cortisol, such as Cushing's syndrome. With fewer osteoblasts at work, the balance tips toward osteoclasts, which remain active. Over time, the bone pile diminishes, and this contributes to osteoporosis and fragility fractures in older adults. The third mechanism is inhibition of withdrawal, which means osteoclast activity is restrained. Here, bone mass is preserved because the rate of resorption is reduced. Weight-bearing activity again plays a role, but hormones are central. Estrogen and testosterone both suppress osteoclasts, which explains why loss of these hormones, menopause in women or hypogonadism in men, leads to accelerated bone loss. Calcitonin, secreted by the thyroid gland, directly inhibits osteoclasts. Adequate intake of calcium and vitamin D are also crucial as it reduces the stimulus for bone resorption. The table provides daily calcium requirements, about 400 to 700 milligrams in children, 1,000 to 1,500 in adolescents, 750 to 1,000 in adults, and even higher requirements in pregnancy, lactation, and after menopause. All of these factors together serve to preserve bone mass by holding resorption in check. Finally, we reach the fourth mechanism, stimulation of withdrawal, where osteoclasts become overactive. This is the pathway that leads to bone loss. Causes include lack of weight-bearing activity as seen in disuse or even in space travel where weightlessness removes mechanical stress. Hormonal conditions such as hyperparathyroidism, hyperthyroidism, hypercortisolism, and deficiency of estrogen or testosterone all enhance osteoclast activity. Acid-base disturbances such as acidosis also favor resorption. Certain malignancies such as multiple myeloma or lymphoma stimulate osteoclasts as well. And of course, inadequate calcium intake and the natural process of aging further tip the balance. The result is a net decrease in bone mass, leaving bones fragile and prone to fracture. So, the level of bone mass remains constant only when the rate of deposition by osteoblasts equals the rate of withdrawal by osteoclasts. This balance may be high, low, or somewhere in between, but it must remain even to keep bone stable. If the scales shift, bone either accumulates or is lost. The table at the bottom reinforces the roles of each cell type. Osteoblasts form new bone, secreting type 1 collagen and matrix proteins. They respond to parathyroid hormone, vitamin D, glucocorticoids, estrogens, prostaglandins, and interleukins. Osteocytes, which make up 90% of all bone cells, are mature osteoblasts, embedded in the matrix. They maintain and preserve bone, sensing mechanical strain and communicating through canaliculi. Interestingly, they can release calcium in response to parathyroid hormone, but are inhibited by calcitonin. Osteoclasts, large multinucleated cells derived from monocyte macrophage lineage, are the resorbers. When active, they develop a ruffled border which increases surface area for dissolving bone. They respond to calcitonin, estrogen, and interleukin signals, but are inhibited clinically by drugs like bisphosphonates. To put this into a clinical perspective, think of osteoporosis. It's essentially the unchecked dominance of mechanism 4, stimulation of withdrawal, combined with reduced mechanism 1, stimulation of deposition. Understanding these 4 pathways gives us a framework to understand not just disease, but also the strategies we use for prevention and treatment: exercise, nutrition, hormone replacement, and targeted drugs. In summary, bone is never static. It is a battlefield of deposition and withdrawal with osteoblasts and osteoclasts at work. The quality of our skeleton depends on which side of this dynamic balance prevails.